All right, welcome to the biology chapter. This will be part one of a two-part lecture. And it looks like the software I'm using to record these lectures simply won't allow me to satisfactorily record the webcam and the microphone at the same time. So you'll just be hearing my disembodied voice off in the ether from here on out. All right, so the mind-body connection. This is something that I briefly alluded to in the very first lecture of what is psychology. And the mind-body connection is this mysterious connection that we don't yet quite have a satisfactory answer for. If you are pressed to give an answer for what is the mind, you realize that it's actually a pretty difficult question to answer. We know that the mind is housed in the brain. That's a good start. This idea of what the mind is probably relates to that squishy, wrinkly pink thing that's inside of our skull. And we know that physical structures influence the mind, they influence mental functioning. So that brain influences our mind. And we also know that mental structures influence physical functioning. So the mind can influence physical functioning. Our mind can move our body around. Our mind can, in a lot of ways, make us feel happy, make us feel sad. So, what is the connection between this intangible mind and this physical, tangible body? That is the root of the issue. How can this intangible thing that we can't quite put under a microscope and study, how does it relate to this thing that we can poke and prod and touch, this thing we call our body? So, a long time ago, there was this idea called dualism. And this idea of dualism is that your body is located in the physical realm and is subject to all the natural laws of nature and physics. But the mind exists in this almost spiritual realm that is not subject to the laws of nature. It's a supernatural realm. And the mind body connection is simply the physical body interacting with that supernatural mind. This is very similar to the idea of a soul. And you can see how that is a little problematic for a science to study something that is supernatural, like a soul. Nowadays, a lot of psychologists who are worth their salt will ascribe to something called materialism. And that is that there is no supernatural plane where the mind or soul resides. There's only the one physical reality that we currently exist in, and that is where the mind and body both are located. All right, so let's talk a little bit about that mind-body connection. Some first attempts to study this were in the field of phrenology. Phrenology is the study of personality via someone's head shape. So these people would look at someone's head shape and via a very intensive, minute study of the different bumps and depressions on someone's skull, they could tell you exactly what type of person you were. This was an idea first made famous by Franz Joseph Gall, who's basically a traveling snake oil salesman. He'd go from town to town with his fancy looking instruments. He would give you a phrenological map that would look something like this. And if you had a little bump in this part, then it turns out you had that particular personality characteristic to a large degree. If you had a little depression in that part of your skull, turns out, well, you didn't really have that personality trait. So he actually wrote entire books based on finding the right husband based on head shape, or looking for a good coworker based on head shape. While this is absolute bogus science and it actually turned out to be quite racist, but the basic idea here is that there is a specific part of the brain that is specialized in a specific mental function. And that idea stuck around, not in anything resembling this phrenology. But so that idea again is localization of function, the idea that a specific part of the brain is responsible for a specific function. Modern day approaches focus on individual neurons. Neurons are individual cells in the brain. Here's a goofy little cartoon picture of one over here on the right side of the slide. There are roughly 100 billion neurons in the mature adult brain. Many different types, many different functions, but this is the basic looking neuron right here. We also have glial cells, and for a long time, literally for decades, we thought glial cells didn't do a whole lot of mental processing themselves. They just simply have a support role to keep these neuron cells alive. We are now finding that is not true, but that is kind of cutting edge stuff that we're not really going to cover in an introductory course. All right, so these neurons, they connect across the brain when they are inside the central nervous system. The central nervous system consists of your brain and your spinal cord. When they're inside the central nervous system, we refer to them as neurons. When they are outside of the central nervous system, we refer to them as nerves. It's kind of a silly distinction, but we also make that distinction between neurotransmitters and hormones. When a particular neurotransmitter is inside the central nervous system, we call it a neurotransmitter. When it's outside, we call it a hormone. We'll talk more about that later on. And these neurons, they're very simple. They are either having an action potential or they are not. An action potential is a very brief, dramatic increase in voltage. So there's a really brief spark of electrical activity, and then it goes back to its resting state. So it has these action potentials all throughout the day when it's functioning. And somehow these tiny little cells that are either on or off create everything we experience in a lifetime as humans. The fact that you are looking at this screen right now and visually registering the words and the colors on this screen means that you have action potentials occurring in the visual cortex of your brain, where those specific neurons relocated to that specific task are located. The fact that you are hearing my voice right now and understanding the words that are coming out of my mouth, that experience of comprehending my speech is all due to millions of these tiny little cells either flicking on or flicking off. That is it. Everything we experience in our world can be boiled down somehow to the action potentials of these very simple little critters. So that is the main question here. How can such simple cells create the human experience? We have incredible complexity from very rudimentary simplicity. All right, so let's talk about these neurons. Let's talk about their basic biological structure. We're going to start with the soma. This is the nucleus of the cell. This is the cell body. This is where necessary cell functions occur just to keep that cell alive. Nothing too exciting there for our purposes. The dendrites are these little branching tree-like structures coming off of the soma, which is represented by this green little smudge here. So the green smudge is the soma. These orange branching structures are the dendrites. And the dendrites receive communication from other neurons. So information is coming into these different dendrites. And then we talk about the axon. The axon is this long, skinny part here of the cell, and this propagates an electrical signal. So an action potential originates around the soma, originates around the dendrites in this upper section of the cell, and it travels down the axon here. Now the myelin sheath is these banded yellow sections surrounding the axon. And the myelin sheath acts as an insulation. It insulates the axon while that electrical charge travels down. So it doesn't lose its charge as it travels down that axon. And last, we have the terminal boutons. The terminal boutons are these branching orange structures at the tail end of the neuron. And the terminal boutons release neurotransmitters into the synapse. We'll talk about that in a little bit more detail in just a minute. But the terminal boutons communicate with other neurons. So the terminal boutons are directly in contact with the dendrites of other cells. So information is coming into the dendrites, an action potential occurs, that electrical charge travels down this insulated axon, reaches the terminal boutons, and neurotransmitters are released into the synapse. Excuse me. All right, moving on. So here's a more detailed image of what we were just looking at. We have the cell body here, the soma. We have the dendrites branching off of the soma. These dendrites are receiving information from other cells. If they receive enough excitatory information, an action potential occurs, and an electrical signal then travels down the length of the axon. The axon is again covered in this myelin sheath, which acts as an insulation. And that action potential reaches the terminal boutons, and they release neurotransmitters into the synapse. The synapse is the tiny little gap between terminal bouton and dendrite of another cell. So the terminal boutons send information, the dendrites receive information. All right, so here in this little simplified diagram, we have one neuron talking to one other neuron. And this is great for sort of explanatory purposes, but in reality, you're going to see something a little bit like this. This is an actual electron microscope image of a slice of brain matter, of your cortex. So the little glowing green balls are the soma of different neurons, the nucleus of those cells. The blue balls are the glial cells that we briefly talked about earlier. And these red branching web-like structures, those are your dendrites, your axons, and your terminal boutons. And I've not actually got a good explanation for what this yellow snaky thing is. But you can see that it's just a jumbled up mess inside of your cortex here. We've got neurons talking to many different neurons. We've got neurons receiving communication from many different neurons. So everybody's talking to everybody, it kind of seems like. All right, so let's talk about that action potential. So action potential is a fancy-dancy physics word. Potential in our fancy physics language just means charge, electrical charge. Action potential means something is happening. There is something happening during this electrical charge. So we have to start by talking about the resting potential. Again, you see that word potential, so that's your electrical charge. And resting just means nothing is happening. When that cell is just sitting there, that neuron is just sitting there, just chilling, it's not receiving any information, it's sitting at about negative 70 millivolts, which is hardly anything. If you were shocked by 70 millivolts, you would not feel it. So the resting potential is negative 70 millivolts. That's what our neurons are just kind of hanging out at. Then we have excitatory communication from other neurons. So neurons are sending information to our cell here. It's receiving it through the dendrites. And lo and behold, we raise the potential. We raise the electrical charge of that neuron by 20 millivolts to negative 50 millivolts. And this is what we call our threshold potential. The threshold potential is where an action potential occurs. So to recap here, we've got our neuron that's just chilling at negative 70 millivolts. That's the resting potential. It's receiving information from other neurons that, hey, you better have an action potential. So the charge, the electrical charge, is slowly increasing. And if it increases to negative 50 millivolts, boom, action potential occurs. All right, so let's talk a little bit more about an action potential. Here's what that looks like in graphical form. So on the y-axis here, we have electrical charge, membrane potential, all the way from negative 100 millivolts up to positive 50 millivolts. On our x-axis, we have time in milliseconds. So you can see our scale goes from before 0 all the way up to 7. So we're talking about a matter of 7 milliseconds for a neuron to have an action potential. Milliseconds are a 1,000th of a second. So 7,000ths of a second is all it takes for a neuron to have an action potential. So here we are, that neuron's just hanging out at our resting potential of negative 70 millivolts. It's getting a little bit of excitation. Other neurons are talking to it, trying to convince it to have an action potential. Boom, it gets to that threshold potential of negative 50 millivolts. And then we have this wonderful little roller coaster here. All right, my mouse keeps disappearing. This increase in charge is what we call depolarization. It reaches a climax and then it quickly repolarizes back down a little bit below the resting potential of negative 70 millivolts. And that little brief period where it's below negative 70 is what we call hyperpolarization, and then it comes right back to the resting potential. All of this happens, again, in a matter of 7 milliseconds. So this is the only thing that a neuron does. It either is having an action potential or it is not having an action potential. And again, we have hundreds billion of these little critters in our brain, and they are either having an action potential or not having an action potential. And somehow our mind can decipher all of that cacophony of information and translate it into everything that we perceive in the world around us. Again, incredible complexity from incredible simplicity. All right. So again, neurons create everything we experience throughout our lifetime. We are either having an action, a single neuron is either having an action potential or it is not. This is an all or none type of situation. There's no variation in intensity. Every single action potential is going to look like every single other action potential. The only exception to this is if the neuron is not functioning properly, if it's damaged in some way. So how do we observe differences in stimuli intensity? Let's say the difference between a very dim light or the difference between a very bright light. How do we observe the differences there? Well, it can't be because we have a really strong action potential or a really weak action potential. Because again, back to the all or none law, every neuron's action potential is going to look like every other neuron's action potential. So one way we can do this is by the number of neurons that are having an action potential and the frequency at which a neuron has an action potential. So let's start with the first one here, number of neurons firing. Let's say we've got 5 neurons firing in response to a light that we're exposed to. Well, that might be a very dim light compared to a light that we're exposed to that causes 100 neurons to have action potentials. Our mind might interpret those 100 neurons firing as a very bright light compared to the very dim light from the 5 neurons having action potentials. And likewise, we can also look at the frequency of how often a specific neuron is having an action potential. If we're looking at one neuron and it's just boom, 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 one right after the other, just constantly having action potentials, well, again, that might be a very bright light. Now, let's say we're exposed to a very dim light and that neuron is just having an action potential now, 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 now. That's not very fast. So our mind might interpret that as a very dim light. So through these two mechanisms, the number of neurons that are firing and the frequency with which that neuron is firing, that is enough information for us to determine differences in stimuli intensity. Now let's talk a little bit more about how neurons communicate with one another. Communication between neurons is not complicated. It is very simple. It falls in one of two categories. It is either excitatory communication or inhibitory communication. Excitatory communication increases the charge of a neuron. Remember, the resting potential is negative 70 millivolts. So if we're increasing the charge of a neuron, we are getting it closer to that threshold potential of negative 50. So excitatory communication makes a neuron more likely to have an action potential itself. Inhibitory communication is the exact opposite. It decreases the charge of a neuron. And if we're sitting at negative 70 millivolts, decreasing the charge will get that neuron farther away from the threshold potential, make it farther away from an action potential. So excitatory communication means that a neuron is more likely to have an action potential. Inhibitory communication means that a neuron is less likely to have an action potential. And that is it. That is the only way that neurons communicate. They're not sending very complicated messages to one another. They're either trying to get their buddies to have an action potential with them, or they're trying to stop everyone from having action potential. Now let's talk a little bit about how that communication happens. So at the terminal bouton, remember that is at the end of the axon, we have a tiny little gap between our terminal boutons and the dendrite of another cell, the dendrite of another neuron receiving the information. This little gap is called the synapse. Now, we have a little bit of terminology here. The presynaptic neuron is the neuron that is releasing the neurotransmitter into the synapse. And we'll talk about that later, but that's how communication happens. Neurons release neurotransmitters into that little gap, and it floats across the synapse onto the postsynaptic neuron. The postsynaptic neuron is the neuron receiving the neurotransmitter. So the presynaptic neuron sends the information, the postsynaptic neuron receives it. Now, inside the terminal boutons, which is the very, very tip of these branch-like structures coming off the end of the axon, we have what are called vesicles. And a vesicle is just a tiny little bubble that contains neurotransmitter molecules. These vesicles are released, they release the neurotransmitters inside of them, and those neurotransmitters, again, they go into the synapse, and they float across that gap, and they fit into what we call receptor sites on the dendrites of the postsynaptic neuron, the neuron receiving the information. And these neurotransmitters fit into specific receptor sites, kind of like a lock and a key. Neurotransmitters are molecules, and they have a specific molecular shape, and each of these receptor sites will receive a different shaped molecule. So all these neurotransmitters have to find the correct receptor sites in order for that communication to occur. All right, so here we have on the left-hand side of the slide, we've got a jumbled up web of dendrites and axon terminals. The red is representing our presynaptic neuron. We've got the axon here, the rest of the neuron is off the screen. And at the end of the axon, we have our axon terminals. And at the very tip of our axon terminals, we've got terminal boutons. And they are all connecting to the dendrites, which are the blue branch-like structures of this cell on the bottom side of the screen. Now, if you look over on the right side of the screen, we've got a magnified image of one tiny little synapse here. So this is what our synapses look like. This big blob here, kind of bell-shaped object, this is the terminal bouton of our presynaptic neuron. This curvy shape here is the dendrite of our postsynaptic neuron, the neuron that's receiving the information. So here in the presynaptic neuron, we've got these tiny little balls. Those are our vesicles, and those little black dots represent neurotransmitters. So the vesicles go to the edge of the presynaptic neuron, they dissolve briefly, and they release the neurotransmitters into the synapse. It's these black dots floating in the synapse here. They float across that synapse, they land on the postsynaptic neuron, and they lock into a shape-specific receptor site and either exhibit excitatory communication or inhibitory communication to that postsynaptic neuron. They either make this neuron, this blue one right here, the one that's receiving the information, they make it less likely to have an action potential or more likely to have an action potential. All right. After that occurs, we engage in the process of reuptake. Reuptake is the process of neurotransmitters being absorbed back into the axon terminal of the presynaptic neuron. Now, reuptake serves a couple purposes. Number one, excess neurotransmitters in the synapse creates problems. We'll talk about some mental illness specific problems later on, but number one, first and foremost, it can desensitize the postsynaptic neuron to that particular neurotransmitter. Let's imagine that dopamine, which is a very common neurotransmitter, is released into the synapse, and you've got all this dopamine floating around, locks onto the receptor sites, and does what it does. It either creates excitatory communication or inhibitory communication on that postsynaptic neuron. Well, if all of that dopamine just sits around and it's constantly trying to lock back into the receptor sites of that postsynaptic neuron, that postsynaptic neuron is going to adapt. It's going to realize, like, hey, there's all this dopamine just floating around in the synapse. So this is the new normal amount of dopamine. I'm going to adjust the number of receptor sites that I have so that it reflects the normal. So now if you need to pass information using dopamine as a neurotransmitter, you have to use an even greater dose across that synapse there. So it changes the amount of neurotransmitter that's required to pass the same amount of information as was before. And the second purpose is a little more pragmatic. It simply recycles neurotransmitters so that our body doesn't have to create more. So that's the process of reuptake. Neurotransmitters are absorbed back into the presynaptic neuron via the axon terminal boutons. All right, let's talk about neurotransmitters. So it's very important to understand that there is no one-to-one correlation between a neurotransmitter and a function. Neurotransmitters have multiple uses, and we're going to talk about a couple very important, very common neurotransmitters that are listed on the screen there. Acetylcholine, dopamine, serotonin, GABA, and glutamate. So let's look at acetylcholine first. Now, again, neurotransmitters have many functions. Acetylcholine is used usually most predominantly for muscle movement. So voluntary muscle movement, involuntary muscle movement, anytime smooth muscles or voluntary muscles need to move, acetylcholine is usually involved. Acetylcholine also plays a big role in learning, also plays a big role in memory. So again, I'm going to go back one slide. Just want to reiterate this phrase right at the top here. There is no one-to-one correlation between a neurotransmitter and a function. Acetylcholine is involved in muscle movement, learning, memory. And what it does is it has an excitatory effect on muscles. It allows our muscles to contract. Moving on to dopamine. Dopamine is probably a neurotransmitter you've heard of. It's very common. It's kind of made a little bit of the rounds around pop culture, I guess, even. But again, a lot of people think, dopamine is like the happy molecule. If you have dopamine in your brain, you're happy. Well, it's not that simple. It's a very nuanced relationship between dopamine and the emotion of happiness. So right off the bat, dopamine is not just involved in the experience of happiness. Dopamine plays a very big role in voluntary, smooth muscle movements. Dopamine also plays a big role in learning, attention, and then lastly, emotion. That's where we think of dopamine being the happiness molecule. But again, it's not like if you're sad, you just take a shot of dopamine and you'll be happy. It's not that simple. It's very, very complicated. And in fact, dopamine is a great example of just how complicated this can be. So, schizophrenia and Parkinson's disease are two mental disorders that, on the surface, look completely unrelated. Schizophrenia is highlighted by hallucinations, delusions, paranoia. You've probably seen schizophrenia portrayed in movies or TV shows, most likely inaccurately, but it gives you the basic idea of what schizophrenia is. Parkinson's disease is very much a muscle movement disorder. People with Parkinson's disease tend to not be able to make very smooth voluntary muscle movements. They have a shake or they have severe difficulties in walking around, and a lot of them might use a cane or a wheelchair. These two disorders are linked through the commonality of dopamine. Schizophrenia is often caused by an excessive amount of dopamine. Just like I was talking about earlier, the postsynaptic neurons sort of adapt to how much dopamine is in the synapse, and they become less sensitive to dopamine if there's an excessive amount in there. So, an excessive amount of dopamine typically leads to schizophrenia-like symptoms. So what does that mean? You just take a pill and reduce the amount of dopamine in someone's brain? Well, that's how you get Parkinson's disease symptoms. Parkinson's disease is characterized by a lack of dopamine, specifically in a part of the brain called the substantia nigra. So again, it's not that simple. It's not like you can just increase the amount of dopamine in someone's brain and voila, they'll be happy the rest of their lives. If you do that, there's a good chance they'll be schizophrenic symptoms. It doesn't mean you can just reduce the amount of dopamine if they're experiencing schizophrenia, because lo and behold, then you have the opposite end of the spectrum, which is Parkinson's disease. So I'm trying to get this idea across that neurotransmitters are complicated. They don't just have a one-to-one relationship between a specific neurotransmitter and a specific function. Let's look at another one. I already talked about this. Okay, so Parkinson's treatment, usually the idea here is to increase dopamine levels. And if someone is suffering from schizophrenia, usually the idea here is to inhibit dopamine production. Obviously, that is not the only form of treatment available for these two disorders, but those are usually ones that are looked at first. Again, this just highlights the idea that there's no one-to-one correlation between a neurotransmitter and a function. More of one neurotransmitter does not always equal better. Dopamine's a great example of that. All right, let's move on to serotonin. Some of the key functions that serotonin plays a role in: mood, hunger, sleep arousal. That should actually be another separate item there: sleep and then arousal. So serotonin also plays a pretty big role in mood, just like dopamine, excuse me, like dopamine does. And you might be familiar with this in the terminology of SSRIs, which are selective serotonin reuptake inhibitors. And I just want to break this big chunky term down and see if we can figure out what it is with what we know already. So selective serotonin means that the pill specifically targets serotonin, and it's a reuptake inhibitor. Remember, reuptake is the process of serotonin leaving the synapse, going back into the presynaptic neuron and being stored in those vesicles. So if a pill inhibits that process of reuptake, that means that whatever neurotransmitter it is focused on, which is serotonin, is not going to be taken out of the synapse. It's going to stay in the synapse and continue to send communication to that postsynaptic neuron. So effectively, what an SSRI does is increases the impact of serotonin. You're basically getting more bang for your buck. You have the same amount of serotonin in your body, in your brain, but the process of it leaving the synapse is delayed, if not stopped altogether. All right, and then briefly I want to touch on GABA and glutamate. GABA is a virtually universally inhibitory neurotransmitter. Glutamate is the exact opposite. Glutamate is a virtually universally excitatory neurotransmitter. So there are exceptions to this, but almost every time when GABA is released into a synapse, it will inhibit the action potential of the postsynaptic neuron. Glutamate is the exact opposite. Almost every single time that glutamate is released into the synapse, it will get the postsynaptic neuron closer to having an action potential. And that's all I want to say about that. All right, let's talk about exogenous substances. These are substances that originate from outside of the body. Another great word for this would be drugs. So we have two broad categories of exogenous substances. We have agonists and antagonists. An agonist is going to increase the effect of a neurotransmitter. An antagonist is going to decrease the effect of a neurotransmitter. All of the drugs that exist in the world today can be put into these two broad categories. All of the psychoactive drugs that exist today can be put into these two broad categories, agonists and antagonists. Now let's use these terms in practice here. So an acetylcholine antagonist, remember acetylcholine is the drug that's heavily involved in muscle movement. And let's say we have a drug that is an antagonist for acetylcholine, which means it decreases the effect of a neurotransmitter. An example of this would be cobra venom. If you are unfortunate enough to be bit by a venomous cobra, that venom is a very powerful acetylcholine antagonist. Some of the side effects of being bit by a cobra are paralysis. If that venom works its way through your body far enough and unfortunately gets into your central nervous system, it'll actually shut down your lungs and your heart. So remember, your lungs and your heart are muscles. They need acetylcholine to flex and function properly. So if there's an acetylcholine antagonist getting into the central nervous system, stopping the heart, stopping the lungs from functioning, then you're not going to have a very good day. So in this way, cobra venom is an acetylcholine antagonist. It reduces the impact that acetylcholine has in the body. All right, another great example of exogenous substances is psychoactive drugs. And oftentimes drugs are agonists, and they function by being molecularly similar enough to the shape of neurotransmitters that they trick the receptor sites on the postsynaptic neurons by fitting into that specific receptor site lock. Now, if you want to have some fun and play around with some mice in a cartoon laboratory, you can click on this link. I'll have it linked below the pages on the Canvas website. You can click on that link and you can kind of play around with giving the mice different drugs and see what it does to their bodies, to their brains rather, via that specific drug agonist. I wish we could do this in person. This is actually a lot of fun to play with. But if you have some curiosity, I would definitely recommend that you click on that. It'll do wonders for illuminating kind of what goes on with different substances in our brain. All right, so let's zoom out a little bit. We've talked at length about neurotransmitters and the individual biology of neurons, but let's talk about the different parts of the brain. When scientists talk about the brain, they typically break it up into three different parts: the brainstem, the limbic system, and the cerebral cortex. It's a good idea to think of these kind of like onions. You know, they're layered on top of one another, and they're layered based on how recently they evolved, and they're also layered based on complexity. So starting with the brainstem, This is sometimes called the reptilian brain. We share this structure in common with reptiles, which is good evidence that it was one of the very first brain structures to evolve if we share it with reptiles. And that's represented by this sort of pinkish, beigeish, tannish, orangish kind of thing down here. There's the brainstem, and then this funky looking thing is the cerebellum jumping off of it. But we'll talk about that a little bit later on. On top of the brainstem is the limbic system, represented by this green smudge here. The limbic system is unique to mammals, and it is sometimes called the old mammalian brain. So at some point, millions of years ago, mammals and reptiles diverged in their evolutionary paths, and mammals developed a limbic system, whereas reptiles did not. On top of the limbic system, in blue, we have the cerebral cortex. This is sometimes called the new mammalian brain. This is the most recent part of our brains to have evolved. If you were to crack open someone's skull and take a look at their brain, this is what you would see, that pink, wrinkly thing sitting just underneath our skull. All right, so let's start talking about the brainstem, that reptilian brain, that most basic, oldest part of our brain. The brainstem is not all that exciting, to be honest. It is involved with things like sleep regulation, keeps our heart beating, keeps our lungs breathing. It's almost exclusively unconscious process. All of the action potentials and mental processing that goes on in here is completely outside of our conscious awareness, which is good. We don't need to know that our brain is keeping our heart beating. Okay, beat. Okay, beat. Okay, beat. That would be incredibly distracting if our brain was sending us that kind of information. Same with our lungs breathing. We don't need to know that our brain is causing our lung muscles to flex and exhale and things like that. So, brainstem, not a whole lot going on. Unconscious processing, basic bodily survival mechanisms, I guess you could say. That being said, this is also a very, very fragile part of the brain. If someone were to experience some sort of trauma on the back of their skull at the base of their brainstem here, this is oftentimes very fatal for these reasons listed right over here. This is how we keep our heart beating and our lungs breathing. Also, in the reptilian brain, we have the thalamus, which is very early sensory processing. All of our senses are routed through the thalamus for that early sensory processing, except our sense of smell. And the thalamus kind of acts like a damper. So all of this incoming sensory information routed through the thalamus, the thalamus kind of helps us realize, okay, should we be afraid? Should we not? It's sort of like a sensor. If something doesn't quite look right, the thalamus throws it out. Smell, since it goes around the thalamus, is sort of an uncensored sense. And this is one of the reasons why smell has such a powerful effect on our emotions, because it's not sort of dampened down by the thalamus. We'll talk more about that when we get to the sensation and processing, our sensation and perception chapter. The reticular formation is another important thing to know about the reptilian brain, the brainstem. And this is where we have arousal, not sexual arousal, but physical arousal. Are we awake? Are we moving around? Are we able to understand the world around us? Or are we just barely sleepwalking, that kind of stuff? There have been some kind of barbaric studies by today's standards where scientists have performed experiments on cats or rabbits, and they'll stick little electrodes into the reticular formation of these critters. And depending on how much stimulation they send, they can make these little critters go absolutely crazy, bouncing off the walls awake, or they can reduce the amount of stimulation and they'll just go right to sleep. The cerebellum is this big chunky thing sitting off the back here. If you could say there is a place where motor memory is stored, this is where it would be stored. So voluntary muscle movements sort of pre-programmed voluntary muscle movements. So things like riding a bike or tying your shoe, things that you don't have to think about when you're doing, that information is stored in the cerebellum. Let's say you're like a professional athlete. Maybe you're a defenseman in hockey and you've taken a lot of slap shots over the course of your athletic career. That information of how to take the perfect slap shot would be stored in the cerebellum. And again, this is all unconscious processing. Good athletes don't have to think about the motor movements required to take highly accurate, very fast slap shots. Moving on to the limbic system, which is the green smudge on top of the brainstem. So the limbic system sits on top of the brainstem. It surrounds the brainstem. This is where we have very old, actually not old, very strong types of emotion and very strong, overwhelming memories occur. So let's talk about a couple structures within the limbic system. First is the amygdala, and the amygdala is involved with fear and aggression. Boy, Freud would just love to know about the amygdala if he was still around, wouldn't he? So the amygdala, you can perform these experiments on mammals where you open up their brain, stick electrodes into the amygdala, and if you stimulate it, you usually get an aggressive response or a fear response. If, however, you are someone who does these types of things, you can lesion the amygdala, meaning you could damage it. If that's done, then whatever critter that is done to will show no aggression, no fear for the rest of their lives until that amygdala is repaired. A great case study is done or was done on a 44-year-old woman named SM. Usually when we talk about case studies, we just use people's initials so that they can keep some anonymity. There's this great case study done on this woman who was born with a damaged amygdala, and throughout her entire life, she never experienced aggression, never experienced fear. Like, can you imagine that? Going through your whole life without feeling any little inkling of fear or aggression. Kind of bizarre, honestly, to think like that. And she tells this story to the neuropsychologist that she was working with. And she just talks about being mugged one day walking down the street. And the person doing the mugging literally had a knife at this woman's throat. And her response to that situation was, well, you'll have to go through God's angels first. Like, what? That is not the appropriate response in that situation. Anyway, way to go, SM. You're brave, I guess. All right. Also in the limbic system, we have the hypothalamus. And the hypothalamus is a wonderful, interesting structure in the brain. Again, we're in the limbic system, so we're dealing with sort of very powerful, basic types of emotions. Now, the hypothalamus plays a role in reward and motivation as well. The 4Fs is sort of a funny little way for people to remember what the hypothalamus does. The 4Fs are fighting, flighting, so that would be your fight or flight response. Food, which would be involved with the reward response. Food is often a very rewarding thing to acquire. So the 4Fs: fight, flight, food, and then the last F is sex. Yeah, I know it's a bad joke. But sex is a powerful motivation for a lot of people, and that motivation is usually housed within the hypothalamus. We're going to talk a lot more about the hypothalamus later on, specifically when we talk about a man named HM. Now, I have another video here that I will link on the Canvas page, and it is of a rat self-stimulating, quote unquote. So this rat has an electrode hooked up to his hypothalamus, and every time he presses a bar in his cell, his little cage there, he gets a little zap of stimulation to his hypothalamus. And rats will quickly learn that pressing the bar is a very pleasurable experience, and they will literally press that bar until they die. Pressing that bar becomes more motivating, more rewarding than eating or drinking. So food and water become less of a reward than simply pushing that lever and getting a direct stimulation to the hypothalamus, which is kind of crazy when you think about it, that rat will literally sit there and die of thirst or die of starvation simply because it wants to keep pressing that lever. All right, so here we have a diagram of the hypothalamus, which is this purple structure here. We have the amygdala, which is this little blue ball right underneath it, and the hippocampus, which sits on top of it. The pituitary gland is something that we're not going to talk about until next lecture. All right, and now we come to the cerebral cortex, the most advanced, newest part of our brain. It sits on top of the limbic system, on top of our brain stem. So the cerebral cortex surrounds the limbic system, and it's usually broken up into 4 lobes. We've got the frontal lobe, the temporal lobe, the parietal lobe, and the occipital lobe, and they look like this. So the frontal lobe is this front part of the brain. The parietal lobe sits right behind the frontal lobe, and we've got this connection right here that we'll talk about in just a little bit. The occipital lobe is sitting here at the back of the brain, and the temporal lobe is right down here, right behind our ears. So temporal lobe, tempo, listening, hearing, ears. It's a good way to remember where it is. And then down here, we have the cerebellum, that structure in the brain stem. So frontal lobe, it's a great name. It's towards the front. Temporal lobe, that's a decent name. Tempo, behind the ear, kind of right behind where our ears sit. And then parietal lobe is one of those you kind of just have to remember. And occipital lobe back here. All right, so let's talk about the frontal lobes. We're going to say lobes, plural, because we have a frontal lobe in our left hemisphere and a frontal lobe in our right hemisphere. So we have 2 frontal lobes. This is where the motor cortex is housed. The motor cortex controls voluntary muscle movement, and it is contralateral, which means your right hemisphere controls the left side of your body, and the left hemisphere controls the right side of your body. That's what we mean by contralateral. So if you get damaged on the right side of your frontal lobes, you will have an impairment moving the left side of your body. Same with the opposite. So if you damage the left side of your frontal lobe, the left side of your brain, you will have impairment moving the right side of your body. So in the motor cortex specifically, more surface area is dedicated to more fine-tuned movement. I've got a wonderful, crazy looking diagram here to help us understand what that means. So this is a cross section of a motor cortex. So you're looking straight on at someone's brain, right at their motor cortex in the frontal lobe. And you've got all this surface area right here. And you can divvy up this motor cortex slice into different parts of the body that are controlled by different specific areas in the motor cortex. So you can go down this list here and you can see toes, ankles, knees, hips, trunks, shoulder, elbow, wrists, kind of moving up the leg, moving down to the hand here. And you can just look at the different sized black bars here, and you can see the parts of the motor cortex where more surface area is devoted to those parts. The hand is huge. It's taking up a lot of surface area in the motor cortex. Look at, you've got the hand right here, and then every little finger has another little section of black bar. So this whole area here is dedicated to fine motor movements in our hand. Compare that to our foot. Look how small the foot is. Look at how little amount of surface area is taken up by the foot. We don't have fine-grained motor movement control over our feet. Yes, we absolutely have motor movement control, but not very fine-grained. Think about what we can do with our individual fingers compared to what we can do with our individual toes. We can write, we can use mouses, keyboards, we can do incredibly intricate things with our fingers, and we can't do those things with our toes. So more surface area dedicated to the parts of our body that require more fine-grained motor movement. Now, look at the face, specifically the lips. This is where we have a lot of fine-grained motor movement as well, just like our hands. And this is for speech, for language. We have to be able to move our lips, move our tongue, move our different articulators in very fine-grained voluntary ways in order to produce speech properly. All right, now I know you guys have been looking at this nightmarish type critter on the right-hand side of the screen. This is what we call the homunculus. The homunculus is a proportionately accurate human if that human was proportioned just like our motor cortex. So the hands are huge because they have a lot of surface area in the motor cortex. The lips and tongue are huge because they take up a lot of surface area in the motor cortex. Everything else is a little bit skinny. Torso, shoulders, arms, legs, feet, toes, neck, everything else is pretty small because less of the motor cortex is devoted to those different areas of the body. So that's the homunculus. I hope that doesn't haunt your dreams tonight. All right, so we talked about the motor cortex and how it's contralateral. We talked about this more surface area dedicated to more fine-tuned movement idea. If you would like to see how we know this through cortical mapping. Again, I'll have this link for you guys below. It is a little gory, it is a little gruesome. You will see a bloody brain. And this is how believe it or not, we were able to map and create that little homunculus guy. Basically, you would go into someone's brain and do some mad scientist stuff. You would send little jolts of electricity and see which part of the person's body sort of had a reflexive movement. So, okay, we send a little bit of electrode here, boom, that moved the person's left big toe. Okay, that part of the motor cortex must be devoted to the left big toe. And you go on down the motor cortex to map the whole human body that way. All right, so that's the frontal lobe. Now we're going to move on to the parietal lobe. And the parietal lobe is where the sensory cortex is housed. The sensory cortex is where we get incoming sensory information from our body. So just like the motor cortex controls voluntary muscle movement, the sensory cortex receives information from these different parts of our body. And it is shaped just like the homunculus. More surface area is dedicated to parts of our body that are more sensitive. So our hands and our lips, very sensitive parts of our body. Lots of surface area in the motor cortex is dedicated to processing incoming sensory information from those areas. And once again, the sensory cortex is contralateral. So information from the left side of our body goes to our right hemisphere. Information from the right side of our body goes to our left hemisphere. That's what I mean by contralateral. All right, and then we're going to finish up with the occipital lobe and the temporal lobe. So frontal, parietal, occipital is sitting on the back here. The occipital lobe is known for the visual cortex. And the visual cortex is where visual processing occurs, conscious visual processing. Once again, it is contralateral. So information from your right eye goes to the left side of your occipital lobe. Information from your left eye goes to your right occipital lobe. And then we have the temporal lobes right here behind the ear. And lo and behold, this is where the auditory cortex is housed. And that is all for the first part of our biology chapter. Thank you very much for sticking with me. I hope you found it illuminating, and those of you that are listening at 1.5 or 1.75 speed, I hope